Hey everyone, we're starting our first chapter on human anatomy and physiology, which involves the blood. And the blood is always a great place to start because the blood is sort of paramount to everything that the cell is doing and everything that your body is doing. The cell is going to have to get rid of some exhaust, and one of the main exhausts that it does produce is this gas called CO2. It makes CO2 as a byproduct of cellular respiration. So as it strips down molecules like glucose and fatty acids and things, the oxygen and the carbon from those atoms gets released as this gas CO2. And it's toxic to the cell. If CO2 accumulates, it becomes acidic, and so the cell has to get rid of it, otherwise the pH will be altered. So it does in fact have to get rid of it. And on the flip side, in order to make energy, one of the main molecules that cells will need is oxygen. Oxygen gets used as the final electron acceptor, so as electrons are whirling down the electron transport chain, they need a place to go at the end, and oxygen, being the electronegative atom that it is, it soaks up those electrons and makes water as a byproduct. So that's why you breathe. Even right now, the oxygen that's going in to your body is going down into your cells and being used as an electron acceptor so that you don't have electrons rattling around in your cell unchaperoned. Okay, in addition to that, you'll need different nutrients and things. These would be like the macromolecules like glucose and the different amino acids and ATP and vitamins and coenzymes and enzymes, things that your cell will need. And then also any other waste the cell produces. So this could be stuff like urea is a waste that gets released. Phosphate groups can be released. There's lots of different other types of waste that the cell can make. Okay, well, to get the oxygen and CO2 into and out of the cell, that comes from the respiratory system. And the nutrients come from the digestive system, and the waste go out via the urinary system and the digestive system. And so you have these different systems that are cooperating together with one another, and the blood is central to them all. The blood is the main medium through which all of these substances will travel. And so it is the substance of life. It is the life-giving fluid. And it's so important that we sometimes say that we all die of the same thing. Regardless of what you die of, we all die of the same thing, and that is a cessation of blood flow. As soon as the blood stops flowing, life ceases to exist. So that's how important it is. It is going to set the stage for a lot of the physiology that happens in the body. So it's just all in all a great place to start in our journey here of human anatomy and physiology. Okay, what the blood does for you, as we mentioned earlier, it transports dissolved gases like CO2, oxygen, nutrients, hormones, and metabolic wastes. Okay, in addition to that, though, it plays a really important role in regulating your body's pH and also the ion composition. So in physiology, we talk a lot about the extracellular and the intracellular ion concentrations. Those are important in determining things like how your neurons fire or how your muscle cells contract. And they have to be kept in pretty specific concentrations in order for everything to work properly. So it's the blood that is going to help maintain those levels, and also for the pH. As soon as you become too acidic, your enzymes and other things might not work quite as properly as they should. So maintaining a proper pH is essential for proper body function. So your blood's going to play that role as well. This is done mostly by the fact that it can carry CO2 away. Carrying CO2 away is a main lever that the body has for regulating pH. Okay, blood will restrict fluid loss. So when you get injured and you start to bleed, you lose fluid, you lose water. And the blood has an innate response where it can clot itself and seal itself to prevent fluid loss. This is a process known as hemostasis, and we'll talk more about it at the end of this lecture. Okay, your blood is also a major defense system for your body. There are these entities called antibodies that are in your blood, And antibodies, they can be found in a lot of different places. They can be found in your tears, in your saliva, in your sweat. Any bodily fluid in the blood is no exception. And their positioning in the blood helps to survey and make sure all the different parts of your body are kept free from invading pathogens. You can think of antibodies as being sort of heat-seeking missiles that will seek and destroy something that is foreign and not supposed to be in your body. Well, these antibodies are made actually by blood cells. There are these cells called lymphocytes that are found in your blood. They start off in your blood anyway, and they help to make the antibodies that help you stay healthy. Okay, and then another main function of blood is it's a major source of thermal regulation. So it's the direction of blood around your body that can help you to reduce your body temperature or increase your body temperature. If you're too warm, almost like a radiator, the body will send blood to the surface of your skin so that air can blow past it and carry the heat away. If you are too cold, your body will actually shunt blood away from the surface of the skin, and it will prevent that heat loss that would normally otherwise occur at the surface of the skin. So it helps to keep the temperature in your body, not losing it. Keep the heat in your body, not losing it to the air. Okay, characteristics of blood, it's slightly warmer than our body temperature. Our body temperature is normally 37 degrees Celsius. Blood is a little bit higher than that. It can have a variable color, and its color depends on how saturated it is with oxygen. Each red blood cell carries about 1 billion molecules of oxygen at max capacity. And so depending on how close to that maximum number the red blood cell is, will change its color. If a red blood cell is not fully saturated with oxygen, then it will be more of a maroon color, or even like a darker purple color. If if blood is saturated with oxygen, meaning all 1 billion of those oxygen molecules are in each red blood cell, then it's going to change the color. It's going to be bright red. Blood is slightly viscous, meaning that it's not just a purely free-flowing fluid. It has a little viscosity to it. Viscosity means how thick something is. So think something like honey would have a higher viscosity compared to water, which would have very little viscosity. And blood, because it does have some things in it, some sugars dissolved in it and other things, it will be slightly viscous. And the viscosity can change depending on how dehydrated you are, which can affect how easy blood is to flow through your body, which can also affect things like your blood pressure and how hard your heart has to work. Another reason to make sure you're always drinking plenty of water, staying hydrated. Okay, the blood is slightly alkaline, meaning it has a pH higher than 7. Remember, acidic pH would be less than 7. Alkaline pH is higher than 7. Okay, when you take blood straight out of a person, it's called whole blood. And whole blood has a couple different parts to it. There's a liquid part to it, and there's a cellular part to it. And you can separate the two by spinning it down on a centrifuge. This is something that's commonly done if you've ever given blood or worked for an organization that receives donated blood. Generally what will happen is you have to actually get somebody's hematocrit before they can give blood. This is very simple to do. I frequently do it in lab. You basically take a small sample of blood, put it in a centrifuge. A centrifuge will spin it, and the centrifugal force of the centrifuge will pull the heavier parts down. And the cellular parts are usually heavier than the fluid parts, so when you do spin something on a centrifuge, the heavier cellular parts fall down to the bottom and separate it from the fluid. And so it would look something like this. This is showing you a tube that has been spun down, and now you can see that there's the cellular parts on the bottom and then the fluid on the top. The fluid part of blood is called plasma, and it's going to be mostly water. There are some proteins called plasma proteins which are dissolved in there, And then different nutrients, things like amino acids and sugars and monosaccharides and things would be in there. And then a whole bunch of various hormones would be in there at any point in time. The collection of red blood cells makes up a majority of the remaining part of blood, and that is called the hematocrit. A typical hematocrit would be, it can vary a little bit, but usually people say about between 37 and 44% of the volume. That would be fairly normal. Whereas plasma is usually more like something like 55%, plus or minus a few percent, something like that. A majority of the blood, more than half, will be plasma. The rest will be cellular. Okay, but of the cellular bits, most of it is red blood cells, but there's a small part of it here that's not red blood cells. These are the white blood cells and the platelets, and they form what's called the Buffy coat. It's really hard to see this on a sample when you spin it down. I've never been able to see this on a small capillary tube. But it is there, and it is cellular contents. The cells are a little bigger, so they're less dense than the red blood cells, which is why they stack up on top, and the red blood cells are pulled more quickly down to the bottom. They're more dense. Also in this layer are these little packets, which are called platelets. Platelets are not cells, but they come from cells. There's this cell called a megakaryocyte, and it has extensions of itself that are constantly entering into the bloodstream, and then the laminar flow of the blood cleaves off the little cytoplasmic packets from the megakaryocyte, and that forms these little blobs called platelets. And the platelets don't have a nucleus. They aren't technically cells. They're just little packets of cell, and what they are used for is making blood clots. So we'll learn more about them later, but they basically are like a little sticky substance that can be used to stick stuff together in the formation of a blood clot. One major clinical application to spinning down blood is that you can use it to detect a variety of ailments. And so a really common thing that's assessed in a clinic is the hematocrit. If someone has a depressed hematocrit, it means that they have too few cells, and we would call this generally anemia. Anemia is any condition in which you have a reduced amount of hemoglobin. So even if you had a proper number of cells and the cells weren't carrying as much hemoglobin or iron, we would still call that anemia. Anemia has less to do with the number of cells and more to do with how much iron is available for binding to oxygen. But generally speaking, the fewer red blood cells one person has, the fewer hemoglobin molecules that they will have, which means the less amount of oxygen their blood can carry. It would be anemic. Anemia, people with anemia, they generally feel tired. They can feel sluggish. Remember, oxygen is used to make energy, and if the carrying capacity of oxygen is reduced, you won't be able to make as much energy. And so you would start to feel tired and lose energy. So that is a major symptom of anemia. Anemia can have a variety of causes. It can be caused by dehydration. There are different genetic disorders that can cause anemia. And it's hard to really know what the major cause is, but it is a very common thing that happens, and very common, especially in women who are pregnant. Women who are pregnant sometimes have to get iron infusions to overcome their anemia, and even my wife wanted to give birth at a birthing center in town, and they were monitoring her hemoglobin levels, and they said you have to be above, I forget what the number was, 8 or 9 or 10, whatever the unit is. She had to be above that level to give birth there, and she was not above that level, so she actually had to get iron infusions, basically an infusion of an intracellular or intravenous infusion of iron. Okay, on the flip side, if somebody had too high of a red blood cell count, it's called polycythemia, and this would be caused by something like dehydration. I think maybe I misspoke earlier. I think I said that this is caused by anemia would not be caused by dehydration. Dehydration would lead more to polycythemia because you're losing your fluid volume, and so the proportion of cells to fluid is decreased. So that could be a case. Other genetic problems could be at hand here. I have sometimes polycythemia, and I don't know if it's because I don't drink enough water or whatnot. We never figured out the reason for it. But it would be an elevated hematocrit count. This is problematic because polycythemia would mean that your blood is more viscous. It is harder to flow. It would mean that the resistance is greater, which would mean your heart has to pump harder to get it to flow through your blood vessels. So drink plenty of water. It's not hard to do. I do a really bad job at that. I drink too much coffee and not enough water. So that's my problem. Okay, so that's a little bit about the composition of blood and how it can change. Those are pretty clinically relevant topics because you'll see a lot of that assessment in the clinic, looking at hematocrit and trying to determine the causes of either anemia or polycythemia. Okay, let's talk red blood cells. These are a majority of what we call the formed elements, or the hematocrit. Those are similar words. Formed elements would be all of the cells, so the platelets, the white blood cells, the red blood cells. The hematocrit typically is just referring to the red blood cells. And red blood cells have this fancy name, erythrocytes. Eryth means red, and then cyte means cell, so red blood cell, erythrocyte. Here's what a red blood cell looks like. In 1 microliter of your blood, so 1 cubic millimeter, it would be 4.2 to 5.5 million red blood cells. You have a lot of red blood cells. They're very small. And so in a cell-to-cell count, they account for about 1/3 of all the cells in your body. 1/3 of all the cells in your body is just your red blood cells. Now, they don't take up 1/3 of the volume of your body because they're so small, but there is many of them. Okay. Generally, they run between 7 and 8 micrometers in diameter, and they have a very large surface area to volume ratio. And the reason for that is that they are flat, and they also have this curvature, which is significant. This is called a biconcave disc. If you were to look at the cross-section of this red blood cell, it's a biconcave disc because both ends of it are concave, which means they fold in on itself. And what this does is it increases the surface area. You know, like if I was to just draw a straight line straight across here, that would be the length of the line. And gas exchange can participate across the surface of the cell. So that would be the surface area for gas exchange or absorption of gas that can occur. If I was to bend the line, it would look more like this. And maybe you can't tell just from looking at it, but if I was to take this line and stretch it out, it would actually probably be more like, you know, this long. So when you curve a line, it increases the length of it, and so adding this extra curve gives us more length to the cell than otherwise would be the case if it was just straight across. So it increases the surface area of the cell, and that's really important because a red blood cell's main job is to onboard oxygen and offload CO2, and it only has a couple seconds to do this. The cell is going to whirl past the lung tissue, and only a couple of moments for the gas exchange to occur. And if this cell was really like cuboid, kind of like this, like if this was the cross section, it would mean that any O2 or CO2 in the cell that's accumulating would have to diffuse all the way across this big diameter, this big cross section of the cell to get to the surface. And it would happen much more sluggish than if it was a flat disc like this, and the O2 could just get right to the surface to participate in the gas exchange. Okay, so that's very important. It's got to be flat to be optimized, and it has to have a nice surface area, and that's accomplished here by the biconcave disc. Okay, red blood cells really only have one job, and that is to carry oxygen. So because of that, they don't need a lot of the other cellular equipment that other cells will have. They don't need mitochondria. They don't need the Golgi apparatus. They don't need the nucleus, the genome, any of that. And so they are anucleate, and because they are anucleate, they don't have any of the other cellular contents. When a cell is anucleate, it means it does not have a nucleus with a genome. And if a cell doesn't have a genome, it doesn't have the building instructions available to help it maintain itself. So it would never be able to keep those other facilities running, like the Golgi apparatus and the rough endoplasmic reticulum and all of those things. That all requires a genome. So the cell can get rid of it. Instead, it packs itself full to the hilt with hemoglobin is the main thing that's found inside of a cell. Okay, red blood cells do not have mitochondria, so they don't actually take the oxygen that they hold on to. Remember, mitochondria is what is the organelle that facilitates aerobic respiration, and that's what requires oxygen. And red blood cells don't have that, so they don't actually participate in aerobic respiration. They don't actually use oxygen, even though they are the main carriers of it. They do have to make a little energy, not much. They're mostly passive oxygen taxis, but there is a little bit of energy that would be required on behalf of the red blood cell, and it can make it through glycolysis. When it does make energy that way, it produces a byproduct, and that byproduct can be toxic to the cell, and so that is why the blood has sort of a shelf life to it. If you take blood and put it in a blood bank, it is only good for so long. If that toxin continues to accumulate because the cells are making energy, eventually, if it's not washed in a circulation system, the blood cells can go bad, so to speak. So that does happen. It can be their shelf length can be extended if they're refrigerated, but blood does have a shelf life to it, and that is partly the reason, is that they have to make energy through this glycolysis mechanism, which has a byproduct that is harmful to the cell. Okay, these are flexible. They can get as narrow as 4 micrometers. So take a look at this. We said that the diameter was between 7 and 8 micrometers. By the way, a micrometer is, I believe, if I'm not mistaken, 1/1,000,000th of a meter. So think about a meter stick, divide that into 1,000,000 parts, and take one of those parts. That's the length of a micrometer. And a red blood cell would be 7 to 8 of those, and they have to fit into capillaries. Some capillaries in your body are only like 5 microns thick in diameter, or wide in diameter. So this cell has to shrink and actually fold in on itself to fit a tube that's smaller than this dimension here, which is pretty extraordinary. Usually when we're talking about a red blood cell going through a tube that small, it has to go through and fit many cells through a tube that small in order for the tube to be of much use to the cell. You know, you have this vast capillary network which sends blood to every nook and cranny in your body, and those tubes are so small, going to even the smallest, most delicate remote places. And lots of red blood cells have to pass through those small capillaries, and so they actually stack in on one another in these sleeves, which are called, this is a great French word. You gotta put your shoulder, lean your shoulder in a little bit when you say it. Rouleau. The red blood cells, they form rouleau. Okay, and this is what they look like. They look like Pringles in a Pringles can. Okay, so red blood cells will stack together like this, and they'll pass through the capillary, a single file, rapidly, and that allows many red blood cells to pass through in a short amount of time. There's a great video here that I have that shows this, and you'll watch as these capillaries open, and then the red blood cells rush through, and then the capillaries close, and then they open again, and the red blood cells rush through, and you can see that they are sort of packing in single file through this capillary. What you're seeing here would be like red blood cells forming stacks through a capillary, and then they're joining maybe like a small venule. A venule would be one of the receiving vessels. And this is probably really slowed down. This probably isn't in a healthy living organism with normal blood flow. But you can see just tons and tons and tons of cells flowing through that small tube. Okay, let's talk hemoglobin. Hemoglobin is the main molecule that is inside of red blood cell, and it's 95% of all the protein inside of a red blood cell. There's about 280 million of them in each red blood cell. So I guess I misspoke earlier. I said there was a billion, but there's about 250 million molecules in each red blood cell. And each hemoglobin has 4 chairs on it, so it can carry up to 1 billion molecules of diatomic oxygen, or O2. This carries almost all of the oxygen in the blood, 98.5% of it. A small percentage of oxygen can just be dissolved in the blood and not be bound to hemoglobin, but by and large, most of the oxygen that you breathe in through your lungs, it diffuses into the blood, and then it grabs a seat right here on the oxygen chair, which are these here, the heme molecules. A heme looks sort of like this. There's an iron right in the middle of it. And the iron is very electronegative. It's attracted, or I guess I shouldn't say that. The oxygen is highly attracted to the iron atom, whatever that means. So the diatomic oxygen that we breathe, which is O2, these O2 molecules, they come in and they are very attracted to the iron, so they bind to it, but the interaction is actually quite weak, and that's important. So the oxygen will bind to the hemes in each of these groups, so every hemoglobin molecule can carry up to 4 oxygens. Hemoglobin is called a quaternary protein because it's actually made of 4 separate proteins which are all sort of linked and tethered together. So we have the beta chain 1, the alpha chain 1, the beta chain 2, and the alpha chain 2. 4 separate polypeptides associated together that forms a globulin, is what it's called, or a quaternary structure of the protein where it has several attached together. And then each subunit of the protein, there is a heme group, and on each heme there's an iron atom, and the iron attaches to oxygen. So when oxygen binds to specifically the iron, these little atoms here that are shown in the picture, it will change the color of hemoglobin. So when hemoglobin has 4 oxygens bound to it, it's fully saturated and it will be bright red. And so if you have a collection of fluid that has many of those bright red proteins, it, as a whole, the fluid will be bright red. If oxygen only had, let's say, 1 out of the 4 hemes holding onto oxygen, that would only be 25% saturated. It would actually be more of a dull purple color. And if oxygen, let's say, on average, all of the hemoglobin molecules had on average 3 of the 4 hemes holding onto oxygen, we'd say it's about 75% saturated, and that would be a slightly more red color. Okay, so you can, by looking just simply at the color of blood, you can tell if it has oxygen in it or not, which is a really cool feature, and it's a super important, clinically significant diagnostic tool. You can use a device which is called a pulse oximeter, and it will literally shoot a light beam through your fingernail and figure out what the color, how red the color is actually, and based upon the color, it will tell you what percentage saturation the blood has. So, true story, I was in Costa Rica and I started having a bout of atrial fibrillation, and I was on the in the ambulance going to the capital city, and my heart rate was going all over the place. It was going 79, 162, 120, 30, jumping everywhere. I would put my put my fingers on my carotid artery, and I would hear my pulse, and it was highly arrhythmic. It would go bum 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 bum, and then not beat at all, and then beat once, and then beat 10 times, and then beat 3 times. It was everywhere. And it was very scary. I remember feeling this tightness in my chest. Felt like I was dying. Felt like I was having a heart attack. And on the way to the hospital in San Jose, I think it is, the capital city of Costa Rica, my wife and I, in the ambulance, we watched the EKG machine flailing from one side of the ambulance to the other as we were going up and down these big mountain switchbacks. But as I was watching my EKG slide across the ambulance from one side to the other, I could see, even though my heart rate was everywhere, I looked at the oxygen saturation and it said that it was always above 90%. So that is the most important thing. Your heart can be arrhythmic, and that's fine as long as it's still pumping blood, and as long as that blood is still being brought to the lungs to collect oxygen, as long as you're still breathing, as long as the oxygen content is high, there is not so much of a concern. So I wasn't so worried about it. You know, in those types of situations, you sort of have to wrestle with the frailty of human life, and but I wasn't too concerned. I could see that my oxygen was at a good level, and that's done just with the beam of a laser through your fingers. So a really important diagnostic tool. If you ever go to the hospital for any type of respiratory thing or any type of cardiovascular thing, they'll put a pulse ox on you and closely monitor your oxygen saturation of your blood, and that is dependent upon how many of these hemoglobin molecules as a whole in your blood are holding on to oxygen at full capacity. Okay, now this is a weak interaction, and it's very important that it is a weak interaction because you want oxygen to get onto the heme, but as soon as the red blood cell is brought to the target site, you know, the tissues of your body, you want that oxygen to jump right off. If the hemoglobin holds too fiercely to oxygen, it won't actually jump off the red blood cell and get to the tissues that need it. So it's really very, very important that the interaction is weak. And it is the accumulation of that BPG, that product of glycolysis, that alters the intensity of that interaction. As BPG accumulates, when a cell is sitting in a bag of blood in the blood bank, it makes it such that the interaction is more strong, that oxygen doesn't get off of the hemoglobin, and then that's not useful blood. The blood might be fully saturated oxygen, but if it doesn't release it, it won't actually do its job. Okay, let's talk a little bit about the life cycle of an erythrocyte. And this is important because there's some clinically significant aspects of this life cycle. So generally what happens is a red blood cell is made in your bone marrow, and the stem cell will turn into this cell called an erythroblast, and that will shed its nucleus and become what's known as a reticulocyte, which is the precursor cell to an erythrocyte. And then it will later enter the blood and then become a red blood cell. You don't have to remember these different parts here, but just know that the reticulocyte goes into the blood, and it's going to travel in the blood for about 120 days. That's about the shelf life of a red blood cell. Again, red blood cells don't have a nucleus, so they can't repair themselves, and eventually they'll undergo hemolysis, which is the breakdown of the red blood cell, and then the body will have to recycle them. Okay, so old and damaged erythrocytes, they're usually phagocytized by macrophages, which are these cells that sort of eat and consume debris in the body. And there will be some in the blood, there will be some in the tissues. Generally, these macrophages are found in the bone marrow, the liver, and the spleen. These are big organs that clean the blood, so to speak. They are always monitoring the blood. Okay, when a red blood cell is damaged and after its long 120-day journey and it's being recycled, the recycled bits go to different spots. Okay, so what will happen is the red blood cell will go and it will fuse with a lysosome in the cell, and it will break it down, and you have a variety of different parts that are released. The globin amino acids and cell components, so the globin would be the globular parts here, the proteinaceous parts, the polypeptides, those are broken down, and the amino acids and the cell components, they're broken down and they're just reabsorbed by the blood to make new red blood cells. The hemes have, in particular, an important fate. Okay, the iron will be recycled. There's a couple of molecules that hold on to iron. Transferrin and ferritin are two that come to mind. Ferritin is sort of what sequesters or holds on to iron inside of the cell. Transferrin, as the name suggests, carries it in the bloodstream to different places. Okay, so iron will likely land there in the hands of either transferrin or ferritin, but the heme that's left after the iron is removed, that has a special fate. That goes on, and it will go on to the liver, and well, first it gets converted into this thing called biliverdin, okay, and then biliverdin will travel in the blood and it will make its way to the liver, and it will then get converted into bilirubin. And this is always where I have to stop and tell you about my friend Billy Rubin. I went to a high school with a kid named Billy Rubin, and great guy. And I always thought it was amusing that his name was Billy Rubin. Okay, but the biliverdin is made into bilirubin in the liver. The bilirubin is going to go down and be stored, or it's going to be converted into bile in the liver, and then the bile has these cells in it called hepatocytes, and they make bile. The bile accumulates in these little canaliculi and then drains down into the ducts and eventually into the common bile duct, and then that will go into the gallbladder, okay? Or it will keep trickling down right in through the hepatopancreatic sphincter into the duodenum. Well, the bile is used to emulsify fats, so it's sort of like a digestive component. So I always like to stop and appreciate how resourceful the body is. It's utilizing these worn out components, recycling into something that's excreted into the digestive tract, but it can still be used to do good. So the bile will emulsify fats, which means takes large droplets of fats, breaks them down into smaller droplets, which can be more easily digested and absorbed. So important thing there. And I always also like to stop and point out that it is the breakdown of bile. So the bile gets converted into these things called urobilins and stercobilins, and those have a brown and yellow color to them, and they are what give your feces and your urine its color. And I always find that that's fascinating, that like when you go to the bathroom next time, you can salute your excrement because it is the color is due to all of the troops of red blood cells that have done their duty and have now come to rest, and those waste products are now being excreted through the digestive tract. Never would have thought that the color in your fecal matter and your urine is from your red blood cells, actually. Okay, some of this will then also get reused to start the whole process again and make new red blood cells. So constantly, every day, you're churning out probably millions of red blood cells. Millions are being made, and millions are being recycled. The heme group goes on to biliverdin, to bilirubin, to bile. Now, I point all this out because bilirubin is sort of a yellowish color, and it can accumulate in the tissues. Once it makes its way to the liver, it's processed to bile. Bile releases it, and it goes down into the gallbladder. But before it gets converted into bile, before it makes its way into the liver and gets converted to bile, bilirubin can be in the blood, and it can manifest in the tissues of the body. And this is a condition known as jaundice or hyperbilirubinemia. Hyperbilirubinemia just simply means elevated bilirubin in the blood. Or this is also jaundice. So people with jaundice, you'll notice a yellow color to the whites of their eyes is typically where you would see it best. I mean, you would see it everywhere, but it's most noticeable in the whites of the eyes, or like the fingernails, anywhere you have sort of a window into something that's supposed to be more of a clear color. It'll be more easy to notice that yellow jaundice. And this is suggestive of some sort of liver disorder. Remember, it's the liver that, it can suggest a couple of things, actually. The liver is the thing that converts bilirubin to bile. And so if the liver is not working properly, something's going awry, then the bilirubin will accumulate, and then it will diffuse into the tissues. It can also suggest a blood disorder. So if there's too many red blood cells being made, and or some sort of infection that's destroying the red blood cells, or some sort of maybe I don't know if this would be the case with like a cancer, like a blood cancer. But if there's too many red blood cells being made and they're being destroyed or they're being destroyed at too great of a rate, that would also really lead to hyperbilirubinemia. So that can tell some important aspects of health there. Babies often, especially if they're premature and their livers aren't fully developed yet, their livers won't be able to process the bilirubin and convert it into bile. And so occasionally, it's not very uncommon, babies will have some jaundice to them. It's sort of fairly normal as their livers are developing. If they're very premature and there's a lot of jaundice, they can treat them with this blue light that sort of breaks apart the bilirubin. As far as I understand it, I don't really know terribly much about it, but that is one of the things that is done in the clinic sometimes. Okay, and then we already talked about anemia. Anemia is another disorder of the red blood cells. Again, a decreased amount of hemoglobin, either because there's fewer cells or because there's just a lower count of iron in the hemoglobin. Usually it takes iron to make hemoglobin, and so if somebody is iron deficient, they also will not be making as much hemoglobin as they ought. They would be anemic. Okay, here's just an interesting look at what it would look like under a microscope if you were to view red blood cells that are anemic. You can see over here the nice robust cells. They appear lighter in the middle because we're looking at that thinner part, the biconcave disc. Okay, this would be like looking at the tops or the bottoms of the red blood cell. And here there's not as much happening inside the cell. They're more translucent. Light passes through easier because there's not as much iron and protein in there to block the light. So this is how anemic cells would appear under light microscopy. Okay, we'll stop there, and when we pick it back up for part 2, it will be talking mostly about the white blood cells, and then we'll also talk a little bit about blood typing.